Hey all, welcome to Share Trek. This is Raj here. And thanks Patreons for supporting the channel continuously. Thanks to Jason, Howard and Eli. You are keeping this channel going. Today I want to talk about mRNA-1644 under the IAVI G003 program. This is another hopeful program that is making progress and has proved quite effective in earlier low-dose trials. So let's examine this a bit more. I'll give you more information. Let's get started. Welcome back, friends. The non-profit scientific research organization IAVI and Moderna announced in May 2022 that the first participant screenings are soon to start for a phase 1 clinical trial of an mRNA HIV vaccine antigen, which is dubbed mRNA-1644, at the Center for Family Health Research, which is called CFHR, in Kigali, Rwanda. And the Aurum Institute in Tembisa, South Africa has also participated in this initiative. The IAVI-sponsored trial, dubbed IAVI G003, builds on the progress in HIV vaccine research. Findings from the phase 1 clinical trial under earlier IAVI G001 showed that the vaccination with the HIV immunogen E0D-GT860MER as a recombinant protein safely induced the targeted immune responses in 97% of recipients, which consisted of healthy US adults. The immune response desired is targeting and expanding a specific class of B cells, which is needed to start the process of developing broadly neutralizing antibodies, which is also dubbed as bnAbs. And the induction of bnAbs is widely considered to be a goal of an efficacious HIV vaccine, and the B cell activation is the first step in this process. IAVI G003 is designed to test the hypothesis that vaccination with E0D-GT860MER or mRNA-1644, developed by scientific teams at IAVI and the Scripps Institutes, delivered via Moderna's mRNA platform, can induce similar immune responses in African population as was seen for IAVI G001 with healthy US adults. Trial sites are expected to enroll a combined total of 18 healthy HIV negative adult volunteers for IAVI G003. All participants will receive 2 doses of EOD-GT860MER mRNA, which contains a portion of viral sequence and cannot cause an infection with HIV. There is no blinding and no randomization in this particular open label study. All participants will receive the intervention. Enrolled participants will be monitored for safety for 6 months after receipt of the last dose, and their immune responses will be examined in molecular detail to evaluate whether the targeted responses will be achieved. The primary trial endpoints are safety and immunogenicity, defined as the ability of a substance to elicit an immune response. EOD-GT860MER was originally developed as a protein by William Schiff, a PhD professor at Scripps Institutes, Scripps Research, and Executive Director of Vaccine Design at IAVI's Neutralizing Antibody Center. The immunogen EOD-GT860MER is designed to be part of an eventual multi-step vaccination regimen that will stimulate an immunoresponse to elicit bnAbs that neutralize or block HIV infection. On their own, EOD-GT860MER will not lead to this outcome, but IAVI G003 will yield important safety and immunogenicity data about this vaccine antigen in a population of healthy adults residing in the part of the world most severely affected by HIV. bnAb stands for broadly neutralizing antibodies, which are a type of antibody that can neutralize multiple strains of HIV, unlike conventional antibodies which only target a single strain of HIV. bnAbs have the ability to recognize and bind to a wide range of HIV strains. making them a promising tool to the development of HIV prevention and treatment strategies. HIV is a highly variable virus, and it can rapidly mutate and evolve to evade the immune system. However, bnAbs can bind to conserved regions of the virus that are less susceptible to mutation, preventing HIV from infecting cells. bnAbs are produced naturally by some people living with HIV, but at low levels and typically after several years of infection. Researchers have also identified and isolated bnAbs in the blood of some individuals who are not infected with HIV, which suggests that these antibodies could be harnessed as a preventative measure against HIV. bnAbs are being studied for their potential use in a range of HIV prevention and treatment strategies, including as a passive immunization therapy, whereas bnAbs are given to HIV-negative individuals to prevent infection. They are also being investigated as a component of an HIV cure strategy, where bnAbs could be used in combination with other therapies to eliminate the virus from the body. In summary, bnAbs are a promising tool in the fight against HIV, as they have the ability to neutralize multiple strains of the virus and offer potential for new prevention and treatment strategies. Moderna is also working on another vaccine, which is called mRNA-1644, which is a HIV trimer. HIV trimer refers to a three-part structure made up of three identical glycoprotein molecules found on the surface of the virus. The three GP molecules are known as GP120 and GP41, and they are linked together to form a complex that is essential for the virus to infect host cells. The GP120 component of the trimer is responsible for binding to the CD4 receptors found on the surface of human immune cells, such as T cells and macrophages. This initial interaction triggers a conformational change in the GP120 molecule, which exposes a site for the GP41 component to bind to the cell membrane. This allows the virus to fuse with the host cell and release its genetic material into the cell, effectively infecting it. HIV trimer glycoproteins have been studied extensively, as they present a key target for vaccine and drug development. Various research studies have aimed to understand the structure, function, and interactions of the trimeric complex, with the aim of developing therapies that can disrupt the trimer and prevent HIV from infecting the host cells. I think I'll make a separate video on mRNA-1644 at a later date, and for now this should suffice. I think we have covered a lot of ground today, of course in a shallow sense, and also the illustrations that I have used in this video are just for visual interest. They are mostly regular antibodies, not broadly neutralizing antibodies. I couldn't find any for bnAbs, but providing some visual interest is important to keep the attention, and that's why I have put it there. So that's all for now today, my friends. I would be back with the next video very soon. I hope you enjoyed this video. If you liked it, please do not hesitate to press a like. And thanks again for your patronage, Jason, Howard, and Eli. Thanks for keeping this going. Bye for now.